The first step in solving this problem was to understand the parasite itself. Ceratamixa shasta is a myxozoan parasite, and the myxozoa are a very large group of parasitic organisms, and most of the nearly 3,000 species infect fish. Until the 1980s, we had very little understanding of how infection occurred, but then the life cycle of one myxozoan was discovered. And instead of the parasite being transmitted from fish to fish, what we call horizontal transmission, it had to first infect an aquatic worm. And this is what we call having a complex life cycle. And this suggested that other myxozoans also had complex life cycles. So our objective was to identify the second host in the C. Shasta life cycle. My interest in the Ceratamixa life cycle began when I was a graduate student, before we had the DNA analysis tools that make research so much easier today. As is often the case in science, we took several wrong turns. For example, early on it looked like freshwater mussels played a role, but by running some carefully controlled experiments, we found we could cause disease even with dead mussel shells. On closer examination of those shells, we found small worms. So that led us back to looking at worms, and we found these small, unusual worms that we learned were freshwater polychaetes. Looking at these worms under the microscope, I saw they had very small parasites in them, and I was sure that that was what we were looking for. But I had to prove it, and without the DNA and analysis techniques, the only way to do that was to put these infected polychaetes in a tank with fish and wait and see if the fish became infected. I started this experiment in November, and over a month later, one of the fish became sick, and when it died, the day before Christmas, it was heavily infected with Ceratamixa shasta. That was in 1996, and it was a very exciting moment for me. I had solved the mystery of the life cycle. The invertebrate host for C. shasta was a polychaete Manianchia speciosa, a worm about the size of an eyelash. Now we understand that there are four major stages of the Ceratamixa life cycle. Infected salmon die and release the myxospore stage. These are small, hard spores. They're shaped like tiny kidney beans, and they'll sink into the sediment. The polychaete worm that lives in the sediment is a filter feeder, and it consumes the spore, which then infects the worm through the gut. The parasite develops in the body wall of the polychaete and is released as an actinospore about a month later. These actinospores are fragile, they look like tiny triangles, and they'll float in the water. That's when they will encounter and infect the salmon, thus completing the life cycle. Our lab and several others had been working independently in the Klamath River, and in 2007 we met to develop a list of management actions that could reduce disease by targeting the parasite life cycle. The goal was to counter the parasite and help to restore salmon populations in the Klamath River. This collaborative group included the U.S. Fish and Wildlife Service Cal Nevada Fish Health Center, the U.S. Fish and Wildlife Service Arcata Office, NOAA Fisheries. We had two collaborators at Humboldt State University, Gary Hendrickson with the Department of Fisheries Biology, and Peggy Wilsbach with the U.S. Fish and Wildlife Service Co-op Unit, and the tribes, the Yurok and Karuks, as well as our own group here at OSU. We developed a list of research priorities for each segment of the life cycle. For actions that reduce myxospores, the questions were: what species of fish contributed most to myxospore production and at what age? How long did the myxospore stages survive? And how many fish carcasses would we need to remove to really make a difference in disease? For actions that reduce polychaetes, the questions were: what is the life cycle of the polychaete and when is it most vulnerable? What habitats are the polychaete found in, and how susceptible are these habitats to disturbance? And what time of year would disturbance be most effective? For actions that would affect infection of the fish, our questions were: how do fish become infected, and would increased water velocity alter the efficiency of the parasite infecting the fish? My laboratory's been working on this problem in the Klamath River since about 2002. First learning where the parasite existed in the river, and then trying to piece together why it's so abundant in certain areas of the Klamath River and whether or not there's anything we can do about it.